Dear learners, welcome to the lecture series of online SWAYAM course on food fundamentals and chemistry. In the last lecture, we learned about the protein. Now it's time to focus on one of the most remarkable function of protein, that is their role as a biological catalyst. Enzymes are specialized protein that speed up the chemical reactions essential for the life. Without enzymes, processes such as digestion, DNA replication and energy production would occur far too slowly to sustain the life as we know it. By the end of this lecture, you will be able to define enzymes, then after that describe the structure of enzymes, then explain the nomenclature system and classification of enzymes, then understand the mechanism of enzyme action, then identify the different applications of enzyme in a food industry including dairy, bakery, fruit juice production, brewing, meat tenderization and so on. Let's dive into the fascinating world of enzyme and uncover the secrets behind the molecular machines that power the life. Let us see what are the enzymes first. So, enzymes play an important role in our life. We know we have already different, we can say products, or we can say different processes are there. In that product and processes, enzymes are involved in the conversion of that whatever the raw material is there into the product. For simple example, we can take from our daily life, that is we will use a curd, that is a yogurt, we can say. In that one the different enzymes are involved. So simple that is conversion of milk into the curd, it is a very common example where this enzymes is utilized. So we will see what exactly enzyme is. So we will define enzymes, like we can say enzyme can be defined as a biological polymer that catalyzes the biochemical reaction. So we can say enzymes act as a catalyst. So what is catalyst? So catalyst is any substance that increases the rate of chemical reaction without being itself changed into the process. Like we can say when we are going to prepare, we can say simple example, whenever we are going to prepare a limbu sarbat, in that one sometime we will add the water, we will add the sugar and we will add the lemon juice. That time we will use the spoon. That spoon initiate or a spoon accelerate the mixing of the that three ingredient. So that spoon act as a catalyst. So here in case of biochemical reaction, this enzyme act as a catalyst and that enzyme increases the rate of reaction. So whatever the most of enzyme, as we have mentioned in the last lecture, the most of the enzymes are three-dimensional globular protein. Specifically we can say whatever the tertiary structure and quaternary structure of protein that is responsible for the enzymatic properties of the protein, or we can say that enzymes are the three-dimensional structural globular proteins are there. So another one important thing is that whatever the enzymes are there, that enzymes convert the substrate into the other distinct molecule and that molecules are called as product, we can say. So basically enzymes convert the substrate into the product. So this is the basic about these enzymes, and enzymes perform the various functions. So let us see what enzymes perform which functions. So enzymes play an important role in signal transduction, that means where the enzymes help to transmit signals within the cells. For example, we can take protein kinase is there that catalyzes the phosphorylation of the protein. So this is about the signal transduction function is there. Then another function is there, enzymes break down the large molecule into the smaller and manageable pieces that body can absorb easily and use easily. So another one very important one function is there, enzymes plays a crucial role in production of energy for the body. So for example, we can take ATP synthase is there, that ATP synthase is an enzyme that helps in creating ATPs and the main energy source of our cells, we can say. Then after that, enzyme assist in moving ions across the membrane, so which is important for various cellular functions including nerve transmission and muscle contraction. So based upon this property, we can say function. The another group is there, which we will see in a further discussion. Then another function is there, enzyme carry a wide range of chemical reaction, such as oxidation reactions are there, reduction reactions are there, and hydrolysis is there. So to remove the waste and non-essential substances from the body. So then another function is there, enzyme helps to reorganize the cell's internal structure to ensure proper cellular function and regulation. So this is all about the protein, how protein is important for the human body, or we can whatever our digestive system is there. In digestive system, starting from mouth to whatever the excretion, all are governed by enzymes. So we have mentioned that in the previous discussion, whatever the part is there, in that one we have discussed the enzyme perform the chemical reaction. So we can see the chemical reactions, all whatever the chemical reactions are there, that reactions either take energy or release energy. So based upon that one, whatever the energy is there, energy diagram is very important. So the whatever the energy of chemical reaction is there, whenever chemical reaction means whatever the reactant are there, whatever we can say reactant A or B, X or Y, whatever that may be. So that reactant, whenever we are combining, mixing, that goes or that convert into the product, but this is a process is there, reaction is there. So for doing this reaction, the activation energy is one concept is there. The activation energy is the minimum amount of energy needed for reactant to form the product in the chemical reaction. So this is activation energy, this is minimum energy required for the, then based upon that energy or based upon heat, the whatever the reactions are there, that reactions categorized into two types, that is exothermic reaction that releases heat energy, that means the energy of product is lower than energy of reactant. So another group of reactions are there, that is endothermic reaction that absorb the heat energy, that means the energy of product is higher than energy of reactant. So these two types of reactions are there, chemical reactions. And what is a relation of the enzymatic reaction with the chemical reaction? So what enzymes perform the role in this one? So can reaction will go without enzymes? Yes, definitely it go without reaction without enzymes, but that time whatever the reaction progress or time of reaction or speed of reaction, we can say that speed is very slow. So how this enzyme will perform their role in the reaction? So simple one, that is enzyme speed up the reaction, chemical reaction, by lessening the activation energy. So we have already seen the activation energy is the minimum energy required to complete the reaction. So whatever that activation energy is there, that activation energy is reduced by this enzyme or by involvement of enzyme. So enzyme does not increase the amount of product formation, or enzyme does not get used in the reaction. So that just speed up the reaction, just catalyze the reaction. So this is about how the enzyme speed up or catalyze the reaction. Then enzyme is, we already seen that enzyme is a protein, and after that enzyme performing the reaction, but we have to know the structure of enzyme, so we can use that enzyme better, in a better way. So the enzyme is comprises of a protein part, that is known as apoenzyme, and another is non-protein part, that is termed as cofactor, or we can say the coenzyme is one of the example of cofactor. The enzyme lacks catalytic activity in the absence of cofactor. If cofactor is not there, the enzyme activity is not there simply. So enzyme together with its protein and non-protein part is called as holoenzyme. So first one, apoenzyme, cofactor, holoenzyme. Then after that, the cofactor may be whatever we have discussed, cofactor, that is non-protein part, that may be organic molecule, and that time it is called as coenzyme, or it may be ion, metal ions is there. When the cofactor is bound so tightly, so we can say that it is difficult to remove without damaging the enzyme. So it is sometimes called as a prosthetic groove. So this is about the structure of enzyme. Then all enzymes have an active site which is specific in shape and specific in functional group for binding to the substrate and catalyzing the reactions. So the active site has two components, that is namely one is a binding site and another one is a catalyzing site. So binding site recognizes the specific substrate and it forms enzyme substrate complex. So this reaction activated catalytic site changes the substrate into the product. So this is about enzyme. Then we can say the enzymes, they have different names, they have, we can say, different classes. So how the nomenclature of enzyme is done. So we know the some of the examples of, we can say, enzymes are there, and so many enzymes are there, number of enzymes are there. So the enzymes have their own system of nomenclature and they are classified based upon the different aspect. Let us see the nomenclature system of enzymes and classification of enzymes. So the common names of the enzymes were derived either from, we can say, firstly, the many enzymes have common names that provide only little information about the reaction that they catalyze. For a proteolytic enzyme is there, that is, we can say, secreted by the pancreas, that is called like trypsin. This is one of the common name of enzymes. So other names, other enzymes are named for their substrate, and we can say for that reaction they catalyze with the suffix of ASE added before the name of enzymes. So this common name pattern were derived either from the nature of substrate they are acted on, or the reaction they catalyzes, or both. So this ended in the suffix ASE. For example, we have taken this one, urease is there. Urease is the enzyme that is acting on urea. Urea is the substrate. So after that urea we have added ASE. So however, this we can say enzyme, we have already seen that one. Sometimes the same enzyme has two or more names. For example, we can say glucokinase and hexokinase in the glycolysis. So these ambiguities are there, and ever increasing number of newly discovered enzymes are there. So however, the number of enzymes, we already said that the known went up, and there is need to formal scheme of nomenclature was felt, and accordingly there is an enzyme nomenclature system in the place. According to the system, and this system, nomenclature system, according to the system, initially the enzymes are grouped into the 6 classes. each class subdivided into the several subclasses which are further subdivided, and every enzyme is characterized by the specific code number. So, this system, whatever nomenclature system is there, that is system developed by, we can say, International Union of Biochemistry, that is Union of Biochemistry established an enzyme commission to develop a nomenclature system, and they have developed this nomenclature, and different classes are there. So, in we can say previously 6 classes are there, that is we will see in detail. The whatever the reactions are there, that reactions are divided into 7 major group right now we can say, and numbered from 1 to 7. So, whatever enzymes catalyzing that reaction, the first group, that is oxidoreductase, is there, and that oxidoreductase perform or to catalyze the oxidation and reduction reaction that transfer the hydrogen and oxygen atom or electron from one substance to another substance. This is oxidoreductase, and example, some of the example of this oxidoreductase, that is 1-dehydrogenase, oxidase, reductases are the example of this group, oxidoreductases. Then another group is there, that is transferases group is there, that is denoted by EC2, that is second group is there, and that second group transferases transfer of a functional group from one substance to another substance. The group may be methyl group, may be acyl group, may be amino group, or may be phosphate group. So, some of the examples of this group, that is transaminase, kinase, methylase, and polymerase. So, the third group is there, that is hydrolysis, hydrolysis group, that perform the hydrolysis reaction, in that formation of 2 products from the substrate by the process of hydrolysis, and the examples are lipases, amylases, proteases, nucleases are the example of this hydrolysis group, that is group 3. Then lyase group 4 is there, and that lyase group perform, we can say, non-hydrolytic addition or removal of group from the substrate, that is carbon carbon bond are there, carbon nitrogen bond are there, carbon oxygen bond are there, and carbon sulfur bond are there, that bonds may be cleaved by this lyase group is there. Then another group, that is group 5th is there, that is isomerases group is there, and this isomerases group can do the intramolecular rearrangement, that is isomerization changes within a single molecule. So, we have seen that one into the different, we can say, isomeric form of the different compound. So, whatever the change in that intermolecular arrangement, that is done by, we can say, isomerase enzyme, and examples are isomerase, mutase are the example of this isomerases, that is group number 5. Then another group number 6 is there, ligases are there, that is EC6 group is there. That EC6 group, that is ligases, join 2 molecule by synthesis of new bonds like carbon oxygen bond, carbon sulfur bond, carbon nitrogen bond, or carbon carbon bond with a simultaneous breakdown of ATP. So, examples are synthetase are there, DNA ligase are there, RNA ligase are there, and then last group, we can say another new recently formed group of, that is translocase are there, that is EC7 group is there, and this group perform the movement of ion or molecule across the membrane or their separation within the membrane, this function performed by the translocase, and the example of this group is quinol oxidase, cytochrome C oxidase. These are the examples of translocase enzymes are there. So, these 7 groups of enzymes are there, and we have already discussed that nomenclature of enzymes, and in that one we have mentioned that there is 4-digit nomenclature system is there. So, we will see that one, those 7 groups were subdivided and further subdivided into the subgroup, we can say, and each enzyme assigned with the 4-digit number preceded by the letter EC for enzyme commission and the systematic name which could precisely identify all enzymes, the reaction that catalyzes. So let us see the meaning of 4-digit in the nomenclature system of enzyme. So the first number shows to which of the 6 group, now we can say 7 group or 7 classes of enzyme that belongs, and second figure indicates the subclass, and third figure gives the idea about subclass, and fourth figure is the serial number of that enzyme in that subclass. So this is a nomenclature system, general nomenclature system. We will take the one example, specific example, so we can get the idea. The formal we can take systematic name of enzyme catalyzing the reaction is ATP glucose phosphotransferase is the enzyme, and this enzyme indicate that it catalyzes the transfer of phosphoryl group from ATP to glucose. So glucose and ATP, D-glucose and ATP is the reaction. Then by using this ATP glucose phosphotransferase, the phosphate, one phosphate is transferred to D-glucose from ATP. So enzyme commission number of this enzyme is 2.7.1.1. So this is 4-digit number. So first digit, whatever the first number is there, that is 2. 2 means that represent the class name, that is transferase we can say. The second digit or second number, that is 7, that represent the subclass, that is phosphotransferase is there, and third number from the nomenclature of this particular enzyme, that is 1, a phosphotransferase with a hydroxyl group as acceptor. So that represented by third digit number 1, and then fourth number, that is again 1, that is D-glucose as the phosphoryl group acceptor. So this is about the how the nomenclature of enzyme is there. Then after that we have to understand how the enzymes is acted, or how the enzymes perform their functions, that is we can say mechanism of enzyme action. So enzyme increases the rate of reaction by reducing the activation energy, and activation energy we know that is minimum energy required to start the any chemical reaction. When these enzymes come into contact with the substrate, firstly it binds with the substrate S and to form enzyme substrate complex, which is a transient product of enzyme catalyzed reaction. Then after this complex is dissociated into the product and unaltered enzyme. So formation of enzyme substrate complex is explained by the two major mechanism. So what are that mechanism? So first one is the lock and key mechanism. So this lock and key mechanism, Emil Fischer created the lock and key mechanism in 1894, which describes how the enzymes interact with the substrate. According to this model, enzyme and substrate have complementary structural properties such as an active site on the enzyme, and that has been pre-shaped to match the substrate. Simply, the substrate must fit correctly into the active site. It must be we can say right size and shape, have charges in the correct place, have the right hydrogen bond, donor and acceptor, have we can say that whatever the substrate is there, that have just the right hydrophobic patches. The substrate can fit into the enzyme's complementary site. Similarly, how the key fits into the lock. So this interaction creates an enzyme substrate complex through the lock and key mechanism, and this lock and key mechanism assume whatever the shape is there, shape of means active site, that is a rigid one that cannot change according to the substrate. So another model is there, that is induced fit model, and this is another mechanism is there, and this model proposed by Coastland in 1960, and it suggests that enzyme active site is flexible one, and initially that is not complementary to the substrate. So when the enzyme substrate approaches, the enzyme undergoes the conformational change to the comfortably fit the substrate, optimizing the active site for the catalysis. Then this whatever we have seen that mechanism of action of enzymes, then we will move to our another portion, that is enzyme utilization in food industry. So as we have seen structure, properties, classification, nomenclature system of the enzyme, and we know the biocatalytic activity of enzyme is the immense importance in food technology. Many reaction catalyzed by the enzyme contributes toward desired attribute of the processed food. At the same time, the number of enzymatic reaction produce undesirable result also. Further, the enzyme may be used in industry as a component of living cell or after isolation in the free or immobilized form. Let us take up some of the example of enzyme in the food industry. So first we will start from dairy industry. In dairy industry, protease is a very important enzyme that act on milk protein and modify structure and solubility properties of the milk and milk product. It also accelerates the ripening of cheese and improve the flavor intensity of the cheese, we can say. Then another enzyme, that is rennet, that is chymosin is there. So this is another enzyme used in the dairy industry to manufacture a cheese. So it is a coagulant which degrade the kappa casein to produce the cheese curd. Then another very important enzyme, that is lactase. Lactase convert lactose to galactose and glucose. Lactose tolerant person have difficulty in digesting this milk and milk product because lactose production in the small intestine is very less. So lactase is used to produce lactose free food product. It also used to make the frozen yogurt creamier and more flavorful. So another enzyme used in dairy industry, that is lipase is there, and lipase is used to break down milk fat and impart the flavor to the cheese. Exogenous lipases are used to create stronger and flavored cheese such as Italian and Romanos. The flavor is derived from the free fatty acid formed when milk products or formed milk lipids are degraded. So these are some of the important enzymes used in dairy industry. So let us move to the brewing industry. The production of alcoholic drink is an important part of beverage industry. As we know, brewing is the process of producing beer and other malt drinks such as ale and stout. The process of producing alcoholic drink involves conversion of complex carbohydrate into the simpler sugar, that is glucose we can say, and after that glucose is converted into the alcohol. The different enzymes are involved in the production process of alcoholic drink. Let us see the important enzymes used in the brewing industry. So that is amylase. It includes alpha amylase and beta amylase, which break down the starches into the fermentable sugar, that is maltose and glucose. This process is essential for producing wort, and that wort is used for making beer. Then another proteases are there which degrade the amino acid and which improve the clarity and stability of beer. Then yeast enzymes are there, that is invertase enzyme converts sucrose into glucose and fructose, while maltose break down the maltose into the glucose. Then glucanases are there that break down the glucans and xylons, which are complex carbohydrate which cause the haze and viscosity of the we can say beer. So then lipoxygenase that contribute to the development of certain flavor in a beer through their activity often controlled to prevent the off flavors. Then beta glucosidase which releases the organic aromatic compounds from the hop and improving the flavor of beers. Then we can move to the bakery industry. So in bakery industry also enzyme performs the various functions. They improve the bread dough and bread quality, leading to the improved dough flexibility, stability, low volume and crumb structure. In baking bread, the preliminary process involves mixing of wheat flour, mainly starch and protein, and yeast and water. Starch gives glucose, maltose and some oligosaccharide. Glucose and maltose are then metabolized by the enzyme into enzymes in yeast, and carbon dioxide is formed. So then glucose oxidase has been used to replace the chemical oxidant and lipases to strengthen the gluten, which leads more stable dough and bread quality. So then move to the fruit juice processing industry. In that one, pectinases are the enzyme that break down the pectin in a plant cell, which helps to extract more juice, reduce viscosity of juice and make the juice more clearer. Then another enzyme in a fruit juice processing, that is cellulases and hemicellulases, that break down the cellulose and hemicellulose in the plant cell, that helps to release more juice and reduce thickness of the juice. Then another amylases are there that break down starch, which helps to make juice clearer. Then oxidases are there that releases natural flavors from the fruits and making juice more tasty and smell better. Then naringnase and limonate dehydrogenase are there, which removes the bitterness from citrus fruit by degrading naringenin and limonene, a better compound found in the citrus fruits, and improving taste of juices like grapefruit juice. Then meat industry, in meat industry the enzymes are used for the meat tenderization, that is we can say proteolytic enzymes, specifically proteases are there. The papain is the most common enzyme used for the meat tenderization. So then another that is in case of syrup manufacturing, the different enzymes are used, that is amylases, alpha amylases and glucoaminases are the crucial in breaking down the starches into the simpler sugars. That is alpha amylases hydrolyzes shorter chain, while glucoamylases further break into the chain down into the glucose. The process is fundamental in the processing of glucose syrup. Beta amylase is there, another enzyme. This enzyme breaks down starch into maltose, a disaccharide composed of two glucose molecules. Maltose syrup is used in brewing, baking, confectionery, and its mild sweetness and fermentability. Then another one, polylinases is there. Then glucose isomerase is there. This enzyme, specifically glucose isomerase enzyme, converts glucose into fructose, that produces fructose used in high fructose corn syrup, and this corn syrup is used in food and beverage product due to its high sweetness level. Then invertase enzyme is there, that invertase enzyme used in syrup manufacturing, that hydrolyzes sucrose into glucose and fructose, producing invert sugar syrup. Invert sugar syrup is sweeter than sucrose and better solubility, making it valuable in confectionery and beverage application. Another one, transglucosidase is there. This enzyme can produce oligosaccharides from glucose, which are used as a prebiotic in a functional food. Oligosaccharide syrup promote gut health and have a lower glycemic index. So this we have seen the different applications of enzymes in the food industry, in a bakery, in a dairy, in a fruit processing, in a syrup manufacturing, in meat industry. So ultimately we can say enzyme plays a crucial role in the food industry by enhancing product quality, improving processing efficiency and reducing cost. Enzymes have numerous and diverse applications in the food processing industry, including dairy, bakery, brewing, juice processing, syrup, animal product processing. They also help in breaking down complex substances, which lead to better texture, flavor and appearance of the food product. Enzyme also enable more sustainable and eco-friendly production processes by reducing waste and energy consumption. Overall, the application of enzyme has transformed the food industry, making it possible to meet the consumer's demand for high quality, nutritious and tasty product. So hope you all learn and gain the knowledge about these enzymes in today's lecture. Thank you.